Today I'll be talking about DNA replication in prokaryotes. DNA is a double-stranded molecule that carries genetic information. It follows a semi-conservative replication method where half of the DNA is from the old molecule and the other half of it is new nucleotides. During replication, DNA goes through 3 steps: initiation, elongation, and termination. Prokaryotic replication occurs in a circular motion rather than linear. This can be either through the rolling circle method or the theta method. Initiation occurs when there is an origin of replication and it gets identified by DNA A, an initiator protein that then binds to that site, and a replication bubble is formed. I'm going to zoom into the replication bubble that is created so that it is easier to see the enzymes and replication occurring. The blue strand is going to be the top strand, going from the 3' to 5' direction, and the green strand is the bottom strand going from the 5' to 3' direction since DNA is anti-parallel. Helicase, represented with yellow triangles, then unwinds the DNA by breaking the hydrogen bonds present between the nitrogenous bases. A replication fork is created while the helicase unwinds the DNA. Gyrase, the black circle, then adds to the back of the replication fork to prevent supercoiling of the DNA strands that are unwinding. Since the DNA is unraveling to the right, the blue strand is the leading strand and the green strand is the lagging strand. To keep the DNA strand from winding up, single-stranded binding proteins bind to the DNA. The white circles represent the proteins. Elongation occurs when DNA polymerase III begins to add new nucleotides to the free 3' OH group. The top strand has a free 3' OH group, making it the leading strand, and DNA polymerase III, the pink square, can easily add nucleotides onto the strand. The strand in red shows the newly synthesized DNA. The bottom strand does not have a free 3' OH group and requires the help of RNA primase. RNA primase, the orange circles, adds RNTP, a primer shown in brown, to provide a free 3' OH group for the DNA polymerase to add nucleotides. The primase adds in sections, which causes Okazaki fragments to form on the lagging strand. DNA polymerase I, the yellow square, then comes in and removes the primers placed by RNA primase, moving in a 5' to 3' direction, and replaces it with the complementary nucleotides in the 3' to 5' direction. However, a few spaces are left open, which are filled by DNA ligase. DNA ligase acts as a glue to fully combine all the Okazaki fragments and seal the nucleotides. In the image, DNA ligase is represented with a black square. This helps create the phosphodiester bonds between all the nucleotides and terminate the replication in that region. However, what if DNA polymerase I could no longer move in the 5' to 3' direction? In that case, the primers placed by RNA primase would stay put and not be removed. The Okazaki fragments would not be combined because DNA ligase cannot combine RNA and DNA together. There would be spaces in between the DNA. The lagging strand would not be complete and the phosphodiester bonds would not be created either, meaning the DNA would never get properly replicated. Hope this helps you understand DNA replication in prokaryotes, and here are my references. Thank you.